Hey everyone, today we are going to start talking about the last of our 4 mandatory New York State labs. So obviously we're still distance learning, so we're going to kind of do this the same way that I did the Beaks of Finches lab, where you watch the video, you follow along, you listen to my instructions and my understanding. There's going to be a couple parts in this video where I ask you to pause the video, look at a picture, and fill in your own information. But first I want to show you the document that you're going to be working on. So the document you're going to work on here is called the Relationships in Biodiversity. It's a fillable. It is in a Google Slides presentation, kind of like the evolution activity we did a couple weeks ago. So same as last time, you're going to find that the pages are actually the backgrounds on the slides for the most part. So each page looks just like a regular PDF, but I've turned it into areas where you can type in it if you need to. So remember that you can zoom in, and then you click to type, so on and so forth, wherever there are blanks. This one has a lot of instructions you're going to notice. We're going to walk through a couple of different things together, but it does have a lot of instructions. Same here, click to type, so on and so forth. The only tricky part about this one is that the last page of this lab is in landscape instead of in portrait. It basically means it's this way instead of this way. Ooh, that's a big mouse. So I could not set it to be the background of this slide. So you have to be really careful not to delete this picture. See how you can highlight this picture? On any other previous page, you can't do that. Here you can. So you have to be extra careful. Here's the deal though. If you do accidentally delete this picture, this is your undo button here, the arrow. So as long as you haven't done so many things that it can't remember what it last did, if I were to accidentally delete it, I could just click undo, and there it is, it's back, okay? So try really hard, that's why I put that note there, try really hard not to delete it, but if you do accidentally delete it, you should be able to click the undo button over here, and you should be able to bring it back fairly easily. So on this one, you also have boxes. This is your data chart for this whole entire thing, okay? So this is your data chart for this whole lab. This lab is basically consisting of 7 mini labs. 1, 2, 3, 4, 5, 6, and 7 mini labs, okay? The other thing I'm going to do here is I'm going to insert a box up here, because usually in class, I have you guys number these boxes or these tests, and they're kind of funny because they don't actually, oddly enough, go in order. So this first one, structural characteristics of plants, is test 1. Let's see. This one is test 2. That one is test 3. They're almost in order, quite frankly. 5. The one that goes wonky is this one is actually test number 7, and this last one is actually test number 6. Now, why they do that, I couldn't tell you. But I like to have those numbers there, and usually I have you handwrite them in when we're doing this lab in class, so that you don't lose track of which test you're on. So in today's video, we are going to look at tests 1, 2, and 3, and I'm going to have points in the video here where I ask you to pause the video and look at the picture that I've left on the screen, and then you can go back to the video and go on to the next part. So before we get started, make sure that you open up your checklist. Remember, I told you to save your checklist someplace safe, so mine is in my Corona Files folder with my stars in front of it. That's the Living Environment distance learning, living environment 2, distance learning checklist. It is on your Google Classroom. It is not in a topic, so it is probably, if it's like mine, it should be right at the top of your classwork page, because it's not in its own topic. So that's hopefully it's sitting right at the top, just like it is for you guys. Okay. So on the checklist, this is going to be activity number 9. And it is relationships and biodiversity, part 1. We'll probably break this up into 3 parts this week. and it is done in a Google slide deck, just like the Darwin Finches one from last week. Okay, so make sure you add that in if you're working to keep track. These check marks should hopefully start to be there, and that will help you to keep track. So, back to the beginning of this lab. It says Botanicurus is a valuable plant because it produces curare. Curare is a compound used to treat certain kinds of cancer. Curare cannot be produced in the laboratory. Botanicurus grows very slowly and is on the endangered species list, so its ability to provide curare in large quantities is limited. Species that are more closely related to Botanicurus are more likely to produce the important substance curare. 3 similar plant species that are plentiful, species X, Y, and Z, may be related to Botanicurus. You will work as a researcher to gather structural and molecular evidence to determine which plant seeds is the most closely related to the hypothetical species Botanicurus. Use this evidence to decide which plant species is most likely to serve as a source of the important substance curare. So, you will need goggles. Well, you won't because you're at home, so you're probably safe not wearing goggles while you watch the video. However, I will wear goggles. I'm probably going to make Jen help me again with this one. So, I'll make Jen wear goggles too. Do not eat or drink. Again, if you want to have some popcorn while you're watching the movie, that's fine. But we won't be eating or drinking while we do this lab. So then it says to perform the following tests and record your observations in Table 1 on page 8 of this packet. So again, page 8 is that very last page way down here. And this whole thing here is called Table 1, which is really big now because I zoomed in on it. But okay, so this is Table 1 right here. So just so you know, when you're referencing the data table, it's all the way on that last page there. So, test number 1, structural characteristics of plants. So, we have these plants here. I'm going to show you on my document camera. We have Botanicurus. Then I have a card for species X, a card for species Y, and a card for species Z. What I'm going to do, I'm going to put these on the projector. I'm going to zoom in to the best that I can. I'm putting it a little off to the side here just because if I put it right underneath, you get the glare from the light because this part is like laminated. But if I put it off to the side, you can still see it, but you don't get the glare or the light is on the soft part that's not laminated. So hopefully that makes it a little better. So here is Botanicurus, okay? I want you to pause the video now. Take a nice good look at this picture. Let me see if I can focus it a little bit better for you. Okay, and I want you to, in that box on Table 1, which I'll show you where you're going to write this, the last page, you're going to click into this box right here, and you're going to type in, you know, whatever you observe. You might want to make the font a little smaller. Maybe make it a 10. Yeah, there you go, because I'll still be able to read that. But it'll be, you'll have a little bit more space. So maybe you're going to write things like how many leaves it has, how many petals it has. Let's scooch this over. Okay, so you are writing that in this box right here. I can make this 2 screens. There we go, that's not too bad. Okay, so go ahead and pause the video and fill in this box right here with your observations. What are the characteristics of this particular plant? Okay, so then for the next one, we're going to take this one out from underneath here. You can see this one is labeled species X. We are going to center that for you. And now you're going to move down to this box here. And you are going to scooch that over so it's not in the light. And you are going to do the same thing here. You're going to pause the video. And you can go ahead and write down your characteristics of species X, which again is this one here, in this box right here. Okay, once you've done that, here is species Y. You can see that it says species Y on it. Here is species Y, same thing. You're going to write your characteristics of species Y right here in this box under test number 1, species Y. Go ahead and pause the video and write down your observations. And last but certainly not least, species Z. Same thing. We'll leave it in the frame here. Go ahead and pause the video when you're ready, and you're going to type in your answers, your characteristics of species Z into this very last box here. Again, you're looking at maybe the colors, the number of leaves, the number of petals, what different kinds of characteristics are you seeing in these plants. Feel free, if you feel like after you've looked at species Z, you want to go back to species XY or Botanicurus, you absolutely can. Just rewind the video and pull up that picture again. Okay, so go ahead and pause the video and take your notes. Okay, so for test number 2, says structural characteristics of the seeds. So you're going to do the same thing here as we just did for test number 1, only instead of looking at the plants themselves, we're going to look at the bottom of the card that I showed you before, and the bottom of the card has seeds. So I'm going to zoom in as far as I can. I might even move the document camera here and try to zoom in. I realize that this is not the ideal setting to be doing this in. But again, you're going to pause the video. You're going to go back down to your last page here of your slide deck where you have the table. And now you're going to be filling in this second column for test 2. So if you look at this picture, you know it's a little blurry. Let's see if I can focus it a little bit more. Okay, that's not terrible. It's not great, but it's not terrible. Okay, so go ahead and pause the video, and you're going to be using your observation skills to write down what you see and what these seeds look like for Botanicurus. Characteristics of the seeds go right into this box. Okay, once you're done with Botanicurus, we'll do species X. You can see that it is species X. Forget that pen mark there. So go ahead and take a look at species X. You're going to put your observations for the seeds of species X right there in that box. Go ahead and pause the video. Okay, and then we'll do species Y, seeds for species Y. Same thing, go ahead and pause the video, and you're going to type your response for what you see for species Y in this box. And again, the last one, species Z, seeds for species Z. Oh, that's not so bad. There we go. Okay, so take a look at species Z, and you're going to write your observations for what characteristics those seeds have here in this last box for test 2. Okay, so that's test 1 and test 2. So test 3, I believe starts, test 3 starts on page 2. That's where the page numbers are. Test 3 starts on page 2. So test 3 is going to be a lot more challenging at home because test 3 requires a microscope, and I don't have a microscope at home. I don't know about you, but I don't have one. Okay, so for test 3, again, no microscope. But you would look at slides for the cross-section of the stems of each of these plants. So a cross section, it's very important you understand what a cross section is. So if I were to show you a cross section of species Z, for example, I would be taking the stem right here, and I would be slicing it. So it would be kind of like if I took this pencil and I sliced it into pieces. If you're, I don't know, like a block of cheese, you might slice a block of cheese. You might slice pepperoni. I like pepperoni. Any of those kinds of things. Even an apple slice, you might slice fruit, and you get just this flat, thin piece of fruit, or piece of pepperoni, or piece of cheese, or piece of pencil, if I were to, in fact, slice the pencil. So that's basically what they do with the stem. They take the stem and they slice it really, really thin, and then they take those little microscopic, teeny tiny little circles that they have, and you're probably not going to be able to see this, but I guess you kind of can. It's right behind my finger. You can see on the slide, if I can get the glare to go away. There we go. It's right behind my finger, and there's a good spot. Sorry if I'm making you dizzy. But that's the cross-section of that stem. So they all look a little bit different, but obviously they look pretty similar when you're looking at them. There's another one. When you're looking at them without a microscope. So what I have found is some wonderful other living environment teacher somewhere in the world, well, somewhere in New York State, obviously, has developed this document here, which it looks like it's a review of all of the state labs, but it does have this page, which is really nice. So on this page, it says, use the lowest magnification on your microscope to examine the slides that show cross-sections through the stems of Botanicurus species X, Y, and Z. Compare the arrangement, circular or scattered, of the bundles of conducting tissue with the specimens. Refer to Figure 1. So there are bundles inside here. They're arrangements of, basically, they're vascular tissue. So they will move water up and down throughout the stem. They will move nutrients up and down throughout the stem. They will move waste up and down throughout the stem. So there are two different types of these bundles. You could have what would be a scattered bundle, which would be if the whole circle, of course it's not going to work, if the whole circle looked like this. It would be all the different bundles are all scattered all throughout the circle, even though you're only seeing half of the circle. Okay? The other option would be the other side here, is sometimes they have what they call circular arrangement of bundles, where they're really only situated just around the outside of the circle. So, if you look at this wonderful picture that this person has created, you can see that, just make sure we can scooch the whole thing over here. You can see they have, here's that same picture from the lab, and they have their example of Botanicurus, which clearly looks like the bundles are where, hopefully you're seeing that. In species X, the bundles sort of seem to be everywhere. Species Y, everywhere. In species Z, again, these darker circles seem to be just around the outside. So, for your data chart, which again is on the last page, all you have to write is the words either circular or scattered. So go down to your last page. You're going to go to test 3. You're going to look at these in this column here and determine whether they are circular or scattered. You're going to click in the box, and that will be your microscopic stem structure. Click, click, click. And I'm going to have you pause the video so that you can see this picture nice and easily, and go ahead and fill that in. Okay, the very last thing we're going to do today before we end this video is back on page 2. Now that you have some, actually let's go back to page 8, sorry about that. Now that you have some data here, you should have all 3 of these columns filled in. 1, 2, and 3. All 3 of these should be filled in. You should now be able to start to see some relationships between these. Remember that the goal of the whole lab is to determine which of these 3, X, Y, or Z, are the most closely related to Botanicurus. So remember, relationships are shown in various ways, mostly through having common characteristics. So if Botanicurus has the same things here as one of these three things here, then my guess would be Botanicurus is most closely related to whichever one of those it is. So on page 2, it says, base your data for structural relationships between X, Y, and Z. Would you hypothesize, which species, would you hypothesize is the most closely related to Botanicura? So again, you're going to tell me right here, is it species X, species Y, or species Z that is the most closely related? Then down here, it asks you to basically back that up with evidence. So explain how the evidence from your data table supports your hypothesis. What information do you have written in that data table on page 8 that says that is probably the reason? So we're not just throwing out any old willy-nilly hypothesis. We are coming up with a hypothesis that is backed up by the evidence. That's your keyword. Okay? It's got to be backed up by the evidence in your data chart. Okay? So that is it for part 1 of relationships in biodiversity. If you have any questions, let me know. I'll be doing my office hours on the day 2 rotational day, which this week is Monday, Wednesday, Friday from 11 to 2, or you can email me anytime. We'll see you all soon.